F214 excretion. Define the term excretion. This is the removal of metabolic waste from the body. What do I mean by metabolic waste? These are any substances which are the result of cellular metabolism. What do I mean by cellular metabolism? These are biochemical reactions that occur in cells. Example, if I was to take a cell, imagine this is a cell, and we've got a biochemical process that's going on in that cell. We have a substance which diffuses into the cell, which we'll call A, which is then involved in a biochemical reaction to make B and C. B is some useful substance which is used up in the cell, whereas C is not, and C, the concentration of C would increase in the cell and would then diffuse out of the cell. It's quite possible that C could be poisonous. It could alter the pH of tissue fluid in the blood, or it could be involved in a back reaction, okay, which would drive this equilibrium reaction in the direction of making A. Okay, so C plus B equals A, okay, which would reduce the useful product B from being produced. So what the body wants to do is get rid of this substance C. Okay? So there, excreted product may be toxic, could shift the equilibrium of metabolic process, or alter enzyme activity, e.g. change pH, or act as an inhibitor. Okay? So in summary, excretion only involves substances that have been involved in a biochemical process in a cell. Excretion shouldn't be confused with elimination. There are certain things that just pass through, for example, the digestive system, that go in one end and come out the other end, but are never involved in any form of cellular metabolism, and therefore we wouldn't consider them to be excretory products. Moving on. Explain the importance of removing metabolic wastes, including carbon dioxide and nitrogenous waste from the body. Let's look at carbon dioxide to begin with. Carbon dioxide is produced by respiration. Remember the material in F214 in the lower 6, and as you know, it dissolves to form carbonic acid, okay, which then dissociates into hydrogen ions, which would alter the pH of tissue. Okay, so we need to remove that carbon dioxide, which we do via the lungs. So the lungs would be considered to be an excretory organ in this respect. If carbon dioxide levels increase in the tissue, that can lead to a condition known as respiratory acidosis, which would be a problem for people with COPD, which is something we learnt about in F212. Moving on to nitrogenous wastes, if we look at the metabolism of proteins, what we find is that proteins are digested in the gut, and they're broken down to amino acids, which are transported to the liver in the hepatic portal vein. Now, we can't store amino acids in the way that we can glucose molecules and fatty acid molecules. And the reason we can't store them is because of the highly alkaline amino group that occurs in the amino acid, the NH2 group, which is here. This group here would be poisonous, so we can't store these amino acids in the liver. So what we have to do is we have to get rid of these amino acids, and we do this by first of all removing the NH2 group from the amino acid. Okay, and so we remove that, and this process is known as deamination. Okay, it's removed and a hydrogen ion or hydrogen atom is added to form ammonia. Okay, so this process is known as deamination. So I can put that over there. Okay, amino group is removed in liver cells by a process called deamination. Okay, the rest of the amino acid, this bit here, okay, can be utilised in the respiratory pathway. Okay, it can be fed into various intermediates in the Krebs cycle and can be used to generate ATP during respiration. So remainder of amino acid is fed into Krebs cycle and generates ATP. The amino group which has made ammonia, okay, the ammonia, as you know, is a very, very soluble substance, okay, and would be very poisonous, would change the pH of the cell, pH of any tissue fluid, would increase pH dramatically, and would be very poisonous. So what the liver does, what the liver cells do, is they convert the ammonia into a less soluble substance and a less toxic substance called urea. Okay, and what I've got down here is the molecular structure of urea, and essentially what it is a carbon dioxide molecule is reacted with two ammonia molecules. Okay, so we have the carbon double bonded to an oxygen here, and then there are two, if you like, amino groups which are also cobbled onto this central carbon atom. Okay, and that's a molecule of urea. So the overall equation, again we don't want to dwell too much on the chemistry here, but the overall equation is that ammonia, in fact two molecules of ammonia, react with carbon dioxide to form urea, and water is also given off. Okay. Now the urea, which is produced in the liver cells, then passes into the bloodstream, where it passes in the general circulation to the kidney, where it's eliminated from the body. And we'll look at the kidney later on. So urea passes into blood and is filtered from the body by the kidneys. So in summary, looking at my original notes here, the nitrogenous compounds are excreted from the kidney. Amino acids cannot be stored. Instead, they are deaminated. This is a key word you need to know, deaminated, which is the removal of the amino group from the amino acid. The ammonia is poisonous, okay, and it is therefore converted to a substance called urea, and urea is less poisonous and less soluble than ammonia. It is still poisonous, and we wouldn't want to have a large amount of urea in our bloodstream, and therefore we get rid of this urea in the kidneys. Okay, the key idea is deamination occurs in the liver, whereas the urea is eliminated from the body in the kidneys. Moving on. We've got to describe, using diagrams and photographs, the histology and gross structure of the liver. Let's deal with gross structure first of all. If we look down here, I've got a picture first of all of what the liver actually looks like. Okay, it sits in the upper abdomen just below the diaphragm, and it's supplied with blood from the general circulation in the hepatic artery, so the aorta feeds blood into the liver here in the hepatic artery. It also, the blood that's been processed by the liver then returns to the circulatory system. This would feed into the inferior vena cava, but also it has a blood supply that comes directly from the gut. Okay, and that it happens in this structure here, or this blood vessel here, which is called the hepatic portal vein. And this brings all the substances which have been digested and absorbed in the small intestine and the large intestine, and will bring it directly to the liver. Also to consider is bile. A substance called bile is produced by the liver, and we'll discuss later how that's produced. But it's stored in a thing called the gallbladder, and it is then released into the small intestine by the bile duct. If I were to represent that in a simple little diagram, just rearrange some of these words. Okay, so I've got the hepatic artery there, providing oxygenated blood from the aorta. I've got the hepatic vein taking blood back to the general circulation, the vena cava. I've got the hepatic portal vein here, which connects the gut to the liver, and I've got the bile duct, which carries bile from the gallbladder in the liver down to the gut. Okay, so that's a simplified diagram of what the actual liver looks like. Okay, so that deals with the gross structure of the liver. If we now go back to this and look at the histology, we've got to go back to the histology, how the cells are arranged, and how the actual liver goes about what it does. If I look at the histology, first of all, if I look at the histology first of all, if I look at the liver under a microscope, what I can see is it's broken down into what we call a functional unit, which we call a liver lobule. So this thing that I'm circling with a cursor at the moment is what we call a liver lobule. Okay, this is a cross section of one, and you can see that in the middle there's a place where there isn't cells, and that is a vein, which is a branch of the hepatic vein, and then radiating out from the central hepatic vein, we can see that there are lots and lots of channels, okay, gaps between cells, which radiate out from the centre. Around the outside, we have collections of other vessels, two blood vessels, branches of the hepatic artery and the hepatic portal vein, as well as branches of the bile duct, which feeds bile into the gallbladder, and we'll discuss what bile is later on. Okay, so that's what a liver lobule looks like. If you're looking at these under a microscope, they're far easier to see in pig livers, because pig livers have a big capsule of connective tissue which separates each of these liver lobules. Not so easy to see in calf or human livers. If I now look at this diagrammatically, i.e. if I take a cake slice type sample of this to show you what it actually looks like in terms of a diagram of how the cells are arranged, I get this type of arrangement here, where we have in the middle, which is equated to this here, we have a branch of the hepatic vein. On the outside we have branches of structures. We've got the hepatic artery here, we've got the hepatic portal vein here, and then we've got a branch of the bile duct here. What happens is that blood arrives in the liver from the hepatic portal vein and the hepatic artery, and then it is funneled into these channels that exist between liver cells. Okay, so these structures here are all liver cells. We call these hepatocytes, and blood from the hepatic artery and the hepatic portal vein is channeled into these conduits or channels, okay, and pass along these channels towards the hepatic vein. These structures, the key word you must know, are called sinusoids. Okay, so the channels that link the hepatic artery, the hepatic portal vein, to the hepatic vein are called sinusoids. Key word. Must have that in your notes. As the blood passes along between rows of hepatocytes, there's an exchange between the blood and the hepatocytes. So substances which are highly concentrated will diffuse along concentration gradients into the hepatocytes. Okay? Let's deal with bile. One of the substances which are produced by the hepatocytes is a substance called bile, which is a mixture of things called bile salts, and I'll talk briefly about what those actually are a bit later on. But as the hepatocytes produce the bile passes through the hepatocyte into these other channels, okay, and these are called bile canaliculi. Again, a key word. This is singular bile canaliculus, plural bile canaliculi, and the bile runs down the bile canaliculus towards the bile duct. Okay. Also, lining the sinusoids are these cells here. Okay, and they're called Kupffer cells. Another key word, Kupffer cells. And what they do is that they're phagocytic cells, and they engulf worn-out red blood cells, and they break down the red blood cells into amino acids, which can be used for amino acid or protein metabolism in the hepatocytes. Iron, okay, which is recycled and is taken back in the bloodstream to the bone marrow to make new red blood cells. But the heme group, the prosthetic group, which is called the heme group, the rest of it apart from the iron, is broken down into a substance called bilirubin, okay, which passes into the bile canaliculi, which then passes along the bile canaliculi, along here, into the bile duct. Okay, so imagine this Kupffer cell here encounters red blood cell, engulfs it phagocytically, breaks it down into amino acids, iron, which it can recycle and use, but the heme group is broken down and made into a substance called bilirubin, which then passes through these cells into the bile canaliculi, which then passes along to the bile duct. All the bile ducts converge into the gallbladder, and then the bile can be released into the small intestine. So, putting some of those words together just to summarise all of that. If I look at here, so Kupffer cells, which are phagocytes, line sinusoids, engulf damaged red blood cells. The Kupffer cells then break down hemoglobin into globin, which is essentially the protein part of the molecule, iron, and the prosthetic group, which is called heme. The globin and iron are recycled, whilst heme is converted to bilirubin, which passes from the sinusoid, where the red blood cell is broken down, into adjacent bile canaliculi. And then the bilirubin is released, along with bile, into the small intestine. And you break down millions of these red blood cells every day. So the activity of the Kupffer cells is very important. So just going back to the original slide here, which you have a copy of. That's in terms of the growth structure. Okay, so the key words that you need to realise what they actually mean. Hepatic artery takes oxygenated blood from the general circulation to the liver. Hepatic portal vein, this links the intestines with the liver and brings all the absorbed food materials from the intestines directly to the liver. Hepatic vein, which returns processed blood back into the general circulation, and you should be aware of the bile duct and gallbladder, which are involved with bile and transferring bile to the small intestine. Moving on to the histology, the functional unit of livers are these things called lobules. Okay, and as it said here, you should practice drawing one and labelling one, so you know what all the key words are. Okay, and the key words are all the ones which are highlighted here. Okay, so the centre of the lobule is a branch of the hepatic vein. We'll quickly flash back to that so we can show you. I've lost it now. So here we are, hepatic vein here, so hepatic vein, and on the periphery we have hepatic portal vein and the hepatic artery and the bile duct. Okay, other key words: sinusoid, it's the channel between rows of hepatocytes, and bile canuliculi, which is also a channel which is delineated by two rows of hepatocytes that feeds bile towards the bile duct. Other key words: Kupffer cells here, these structures here, these phagocytic cells which engulf damaged red blood cells. Moving on to the next slide. So what you've got to do is describe the formation of urea in the liver, which we've already mentioned, including outline of an ornithine cycle, which is obviously something new to us. So as I said before, we can't store amino acids in the liver. Okay, what we do is remove the amino group. Okay, that's a process called deamination. Okay, key word as I mentioned before. Now the resulting ammonia is then converted to urea in a biochemical cycle, a bit like other biochemical cycles we've come across, such as the Krebs cycle, the Calvin cycle, and so on. It's a cycle of chemical reactions which are involved in producing things. Okay, in this case it's a cycle which is an input of ammonia and carbon dioxide into the cycle and an output of urea. Okay, and we'll just go through that in a little bit more detail. Okay, so as I said, ammonia essentially reacts with carbon dioxide. Okay, and it's a complicated cycle, and I'm not sure whether you actually really have to know this, although this diagram I've lifted straight out of the A-level textbook, so I suspect there is an expectation for you to know this. You should certainly be able to understand it. Okay, as I've got here, I've got a rather awful mnemonic, okay, which helps me remember the order in which everything comes, and I'll explain that in a second. So, as we said, ammonia is produced by the process of deamination, and what happens is that ammonia is fed into this cycle it reacts with a substance called ornithine. Okay, and ammonia and carbon dioxide both feed in to this cycle. So the carbon dioxide feeds in, and the ammonia feeds in, and they react all together to form a molecule called citrulline. Okay, and citrulline is then converted to a substance called arginine, but another molecule of ammonia is also added to convert the citrulline to ammonia. Okay, and the arginine is then converted back to the starting substance, ornithine. But in doing so, we end up producing a molecule of urea. Okay, and that's called the ornithine cycle. Okay, I have a silly mnemonic: old and crusty Charlie, awful ammonia equals urea. So that's how I remember it. It's a rather silly mnemonic, but it works for me. Moving on. Now what we need to look at is the various roles of these hepatocytes. Okay, so we've looked at the structure of the liver, we've found out how the hepatocytes are arranged in liver lobules and how they are able to absorb materials which are in the blood. And now what we want to actually find out is some of what actually these liver cells actually do. We've already looked at how it deals with ammonia from deamination, converts it into urea. But now we need to look at one or two other examples. First of all, liver cells are rich in various enzymes, and some of these enzymes are involved in converting toxic substances into less toxic ones. The example you've just seen of ammonia being converted to urea is an example of that, but it's not the only example. You've probably often used the enzyme catalase, which you've probably found in liver, to convert hydrogen peroxide into oxygen, which is an enzyme assay we often use for working out the effect of temperature and substrate concentrations, etc., on enzyme activity. Okay, you might ask, why on earth is hydrogen peroxide involved in cells? And the answer is it is produced in cells during fat metabolism. So a lot of hydrogen peroxide is produced in cells, and as you know, it's a strong oxidizing agent. It's the substance that you use to bleach your hair, hence the term peroxide blonde. So it's obviously quite a poisonous substance, and it has to be got rid of incredibly quickly in cells, otherwise it would do considerable damage to cells. So there are enzymes in liver cells which convert hydrogen peroxide very quickly into water and oxygen. So that happens in hepatocytes. So just summarising that. So hydrogen peroxide is a metabolic waste. And so hydrogen peroxide broken down to oxygen and water by the enzyme catalase. There's an example there. Moving to other examples. Ethanol is a poisonous substance, and as we know, and is broken down in the liver by an enzyme in hepatocytes called alcohol dehydrogenase. And what that does is it oxidizes the alcohol and uses some of the molecule, or breaks it into acetyl groups, and feeds those acetyl groups into the Krebs cycle. Involved in the link reaction into the Krebs cycle, those acetyl groups are fed in for respiration. So some of the molecule yields ATP. Okay. And I've also written here, too much alcohol reduces oxidation of fatty acids in the liver, which then deposit in the liver to produce fatty liver. These acetyl groups, which are produced by the breakdown of alcohol, are also produced by the breakdown of fatty acids, and these acetyl groups are fed in to the Krebs cycle. So if you immerse your alcohol dehydrogenase enzymes with alcohol all the time, what happens is that you don't break down fatty acids, and therefore that fat is stored in the liver, and that's called fatty liver disease. It's also, if you're on a diet, one of the things they say you should never do is drink alcohol, and that makes sense, because if you're drinking alcohol, you are producing these acetyl groups, which are being fed into the Krebs cycle, which means that you're not breaking down fatty acids. And obviously you're not breaking down fatty acids, you're not using fat, and therefore you're not going to lose any weight. Okay, so that gives you a sort of biochemical reason why you shouldn't drink alcohol if you're trying to lose weight. So, in summary, the ethanol broken down in liver cells by an enzyme called alcohol dehydrogenase. Acetyl groups are fed into Krebs cycle via acetyl coenzyme A, which you'll come across when you study respiration. The next topic is the kidney, and a bit like the liver, we first of all need to look at its gross structure before looking at the functional units of the kidney, which are called nephrons, which you may remember from GCSE. So dealing with its gross structure, here's a nice diagram which shows aspects of the gross structure of the kidney. The kidney if you do a cross section of the kidney, it can be divided into various regions. There's an outer layer here which is called the cortex, and then there's an area inside the cortex which we call the medulla. The kidney is supplied with blood from the aorta and the renal artery, and then blood that's been processed by the kidney then returns to the bloodstream in the renal vein. The filtrate that's produced by the kidney is funneled by a region called the pelvis of the kidney, which is here. It's funneled into a tube called the ureter, which takes the urine, as it's called now, to the bladder. As an aside from one of the last topics we did, sitting like a little woolen hat on top of each kidney is the adrenal gland. It also has an outer region which is called the adrenal cortex and a middle region which is called the adrenal medulla. The adrenal medulla being responsible for producing adrenaline, whereas the adrenal cortex produces corticosteroid hormones, which we studied in the hormones topic. Here's another diagram which shows much the same, except in this diagram it orientates where our functional units of the kidney are. Functional units of the kidney are called the nephron, which you may remember from your GCSE study. But just going through the cross section here, again we have the cortex, renal cortex as it's known, which runs out around the outside. Okay, and then we have the medulla region, which is in the middle around here, and as I say, the renal pelvis, which is where all the filtrate from all the nephrons drains into this renal pelvis area, which is then funneled into this structure here, which is the ureter, which takes the urine off to the bladder. Now the functional units of the kidney, analogous to our liver lobules that we looked at in the liver, in the kidney the functional unit is called a nephron. And there are millions and millions of these nephrons in each kidney, and they're arranged such that part of the nephron can be found in the cortex, okay, and then bits of the nephron stretch down into the medulla, and eventually the filtrate that's produced by the nephron empties through the medulla, this structure here called the collecting duct, into the renal pelvis. And then all the millions of nephrons empty their filtrate into this renal pelvis, which then funnels it into the ureter. Here is a nephron itself, okay, with some structures which you might remember from GCSE. I've got another diagram which will look at this, which perhaps makes it easier to understand, but we've got a knot of capillaries here, which is called a glomerulus, a structure called the Bowman's capsule, a squiggly tubule here, which is called the proximal convoluted tubule. Convoluted means squiggly or windy, proximal means close. And then we've got a long tube which stretches down into the medulla. You can see that on this diagram here, but on this diagram here that's what it is there. It's called the loop of Henle, and for the purposes of this we talk about a descending loop, limb of the loop of Henle, and also an ascending limb of the loop of Henle. We'll look at that in a little bit more detail. And then we have a thing called the distal convoluted tubule, okay, which is also a winding tubule, which is distal, further away from the start of the nephron, and the distal convoluted tubule then connects with a collecting duct, and the collecting duct also passes through the medulla and empties any filtrate into the renal pelvis. So here's the collecting duct here in this diagram, running down and running into this region here, which is called the renal pelvis. So looking at the nephron in a little bit more detail here, here's what you need to know. You need to know the key words, word glomerulus here. You need to know this word glomerulus, which is a knot of capillaries. And the knot of capillaries is in association with a sort of blind end of a tube, which is called the Bowman's capsule, which I'll show you that in a minute. And then the Bowman's capsule leads into a thing called the proximal convoluted tubule, which leads into the loop of Henle, okay, which has got a descending limb and an ascending limb, okay, and then the loop of Henle leads into the distal convoluted tubule, which leads into collecting ducts, which empties into the pelvis, which the pelvis funnels urine into the ureter. Looking at that diagrammatically, I'll just quickly go through that again. So here's a schematic diagram of what one of these nephrons looks like. So we have this knot of capillaries here, which is surrounded by this blind-ended tubule, which is called the Bowman's capsule, and that leads into the proximal convoluted tubule here, which then leads into the descending limb of the loop of Henle here, which then leads into the ascending limb of the loop of Henle, which then leads into the distal convoluted tubule, which then leads into the collecting duct here. Looking at some of these areas in a little bit more detail, we'll move on to the next slide, which describe and explain the production of urine with reference to the processes of ultrafiltration and selective reabsorption. Okay, so there's quite a lot of detail we need to know here. This is a rather elegant diagram here of what's going on in the Bowman's capsule. So first of all, we have a network of capillaries, okay, which are covered in these funny purple cells, which I'll talk about in a minute. But the network of capillaries, or glomerulus, okay, which or glomerular capillary, which is tend to call it the glomerulus, is fed by an arteriole. So the arteriole supplies blood to this network of capillaries, okay, or glomerular capillaries, and the whole thing is called a glomerulus. You'll notice that once the blood has passed through the glomerulus, it then joins with another capillary, okay, and we call that, well, not a capillary, sorry, wrong word, it's an arteriole, and we call that the efferent arteriole. Okay, so it's rather odd in that we have an arteriole feeding a capillary, which feeds another arteriole, because this usually feeds a venule. But in this case it's an arteriole, and we call these, the incoming one, we call the afferent arteriole, and the outgoing one is called the efferent arteriole. Now, one of the key things to notice here, if you notice the width of this afferent or incoming arteriole, it is wider than the width of the efferent arteriole. What this means is that blood being pumped under ventricular pressure or systolic pressure through here, as it goes into, there's more blood coming in here than there is blood coming out of here. What happens is that that creates a higher pressure inside the glomerular capillaries than would normally be the case in a capillary bed. So there's high pressure in this capillary. Okay, and that is one of the key influences in the whole process of forming urine, is that we have, it's a filtration process, and I'll come to the filtration process in a minute, but this word ultra basically means under pressure, and the pressure is created due to the blood pressure pumping blood into the capillaries and the fact that there is a differential between the width of the incoming arteriole, that's wider than that. Okay, so pressure develops ultra in this capillary space here. Okay, so that's the ultra bit of the word ultrafiltration explained. Now, the complicated bit to visualise, or this diagram does actually represent it rather well, is the idea of the filtration. The Bowman's capsule is constructed that it's got an outer layer of cells here, which are normal flattened squamous epithelial cells, which form a watertight structure around the outside of the Bowman's capsule here. The inner lining of cells are quite interesting, in that the inner lining of cells are continuous with the capillaries. Okay, and here's one of these cells here, and these cells have got a special name. They're called podocytes. Podocytes, okay, as outlined there. And the podocytes wrap themselves around the capillary. And I always think of the analogy of a podocyte being a little bit like a hand wrapped around, let's say, a tennis racket. And essentially the key thing is that there are gaps between the fingers, okay, these finger-like projections, which are actually called pedicels, okay, and it's the gaps between these things which are actually the important bit, because that allows easy movement of molecules from the bloodstream into the Bowman's capsule, which surrounds the capillaries. Because if it was a continuous layer of cells, if it was squamous epithelial cells wrapped around these capillaries, it would be very difficult for any of the filtrate from the blood to actually pass into the nephron. So the gaps created by these podocyte cells allows easier filtration. If we were to look at that in a little bit more detail here, we have blood coming along a capillary through here, and there are three things to consider, three layers which separates the blood in the capillary from the filtrate in the Bowman's capsule. The first thing to consider are the endothelial cells of the capillary itself. Okay, and like the capillaries we studied in F214 with respect to tissue fluid formation, these endothelial cells have got pores in them, they've got holes in them. Okay, sometimes the word fenestrated is used. They have little windows in them. Okay, and these little windows enable molecules to pass out of the capillary. Remember, they're not big enough for big cells, blood cells, to be able to pass out of there. Okay, but they are large enough for molecules such as water and oxygen and so on to pass through these fenestrations or pores. Wrapped around the endothelial cells of the capillary, there is a structure called a basement membrane. Now, this basement membrane is a confusing word because one thinks of membranes as phospholipid bilayers in biology, but this membrane isn't a phospholipid bilayer. It's made of a meshwork of collagen fibres. And these collagen fibres act as a filtration device. So they don't allow large molecules or cells to pass through, but they do allow smaller molecules such as water, salts, urea, oxygen and so on. They do allow those molecules to pass through, but they don't allow the larger molecules to pass through. And then surrounding the basement membrane we have these cells which form the lining of the Bowman's capsule around the capillaries, which are these special cells called podocytes, okay, which I made, remember, this analogy about a hand with fingers gripping a tennis racket. So they wrap round the capillary, and it's actually the gaps between the fingers, if you like, or they're actually the fingers are called pedicels. But it's the gaps between these which are the key thing. Okay, they form what we call filtration slits, which allows molecules that have passed through the basement membrane to easily pass into the Bowman's capsule. So, just summarising all of that on the notes that you have. Okay, so you've got to understand what the term ultrafiltration means. The word ultra is to do with pressure, and that's caused by the incoming arterial having a wider bore than the outgoing arterial, as I've just shown you on the diagram. Filtration basically means that the small molecules, water, salts, glucose, amino acids, urea, etc., can leave the capillary in the glomerulus and pass into Bowman's capsule. Okay, and there are three things to consider that these filtered substances that I've just outlined here have to pass through three layers, if you like, between being in the blood and actually getting through into the filtrate in the capillary. The first thing are pores in the capillary endothelial. So the capillary is made of cells called endothelial cells. You should remember that from F214. And these have pores in them. Okay, that word fenestration I used refers to the pores in these endothelial cells of the capillary. Wrapped around the endothelial cells is this structure called the basement membrane, which is like a meshwork of collagen fibres, which acts as the filtering membrane, which prevents large molecules and cells, etc., passing from the blood into the filtrate. Then wrapped around the basement membrane are these specialised podocyte cells. Okay, and the gaps between podocyte cells create what we call filtration slits, which allow filtered substances to easily pass into the Bowman's capsule. The next bit to look at is what we call selective reabsorption. This is quite complicated. This occurs in the proximal convoluted tubule, which is the first of the winding tubules that passes from the Bowman's capsule. If I look at this diagram here, if you can imagine the proximal convoluted tubule, which we saw on the last slide, has the filtrate passing down here, okay, and the proximal convoluted tubule is made of a succession of these types of cells here on either side. So there'll be cells here, cells over here, and the filtrate runs down here. What happens is, as the filtrate passes down, substances that we don't want to lose in our urine, such as glucose molecules, amino acid molecules, etc., water molecules even, are reabsorbed back into the bloodstream. So they pass through the cells that line the proximal convoluted tubule, and they pass through the back end of that cell into the bloodstream. Okay, so a lot of the molecules that we've just lost from the bloodstream are very quickly reabsorbed. But the cunning thing about all of this is that it's selective, is that we only reabsorb the molecules we want, and we don't reabsorb the molecules we don't want. So if we look at this cell, there are various features that we need to consider with this cell. First of all, you notice that we've got these structures here which increase the surface area of the cell in contact with the filtrate. These are called microvilli, which we've come across before. So their job is to increase surface area because we've got an exchange surface here, and therefore we want a large surface area for that to exchange things with. These cells, I'll have to draw these in because they don't appear in this diagram, but these cells would also have lots of mitochondria in the cells because they're heavily involved in active transport, which we'll see in a second. So what happens is that molecules that we want to absorb are absorbed by a special type of transport, which you didn't study at F214, and I'll quickly explain it. It's called sodium cotransport. And what happens is that the molecules, be they glucose or amino acids or whatever they are, we've got glucose here as an example, are absorbed into the cell along with a sodium ion, okay, quite a heavily charged sodium ion. This helps change the conformation of this carrier molecule, which helps pull the glucose molecule through the membrane. So the glucose and the sodium pass through together by a process called sodium cotransport through one of these cotransport molecules here. So the glucose gets into the cell, as does the sodium. And what we do is the sodium is pumped by active transport out of the cell here. The idea being that there's always a steep concentration gradient of sodium in the filtrate and sodium in the cell, which means the rate of diffusion through here is going to be greater because of the higher concentration gradient. The glucose passes by facilitated transport through a glucose carrier molecule out of the other end of the cell too. Other features of the cell, you'll notice that the cells are connected to each other. So the cells here and here are connected to each other. Okay, those are called tight junctions between cells, and that's to prevent any leakage of water or molecules between the blood and the filtrate, or the filtrate and the blood. So it produces, if you like, a fairly waterproof layer of cells, so that molecules have to pass through these special carrier molecules. And there will be different carrier molecules for different things. So there would be each amino acid, for example, would have its own specific carrier molecule. Each of the 20 different amino acids would have specific cotransport proteins which would take those amino acids into the cell along with sodium ions. So that's how that works. It's quite complicated. Okay, so I'll just quickly go through that again. So the microvilli, just using this as a prompt, the microvilli, increased surface area. Okay, the membranes have these cotransport proteins which take molecules such as glucose or amino acids into the cell along with sodium ions. Sodium ions are then pumped out actively to lower the intracellular sodium concentration, which means there's a steep concentration gradient between the filtrate and the cell, which means that there's a higher rate of diffusion of sodium into the cell along with the glucose. Glucose passes out of what's called the basolateral membrane, which is the back end of the cell, which is associated with the blood capillary here. So the glucose passes out of that into the blood capillary. So what we've done is reabsorb glucose from the filtrate back into the blood here. I've also pointed out that these cells have lots and lots of mitochondria because they need to produce lots and lots of ATP to be able to pump these sodium ions into the blood. So the next thing we have to look at is explain, using water potential terminology, the control of water content of the blood with reference to the roles of the kidney, osmoreceptors in the hypothalamus and the posterior pituitary gland. Okay, so we just looked at in the previous slide of how a number of substances such as glucose, amino acids, are selectively reabsorbed from in the proximal convoluted tubule. In actual fact, lots of water and salts are reabsorbed in the proximal convoluted tubule, but there is now a special mechanism which enables us to absorb more water and more salts from the kidney filtrate. So, things to consider here is that In the descending limb of the loop of Henle, water passes from the tubule to the medulla cells. These are the cells surrounding the loop of Henle. Okay, and that happens purely by osmosis, simply because the water potential in the filtrate is greater than the water potential in the cells, or the tissue fluid that surrounds the cells of the medulla. So that's what's happening in the descending limb on the way down. And on the ascending limb, what we do is we pump sodium and chloride ions out of the filtrate, okay, which actually reduces the water potential of the medulla, which draws water out in the descending limb. I'll show you that in a second. So if I look at this diagram here, just to summarise what I've just been saying. So this is the descending limb, and as we can see, as the filtrate passes down the descending limb, water moves out of the filtrate. The reason it moves out of the filtrate is because the water potential is higher in the filtrate than it is in the surrounding cells. So as the water moves down here, as you can see, it becomes progressively more concentrated. So 300, whatever that 300 is, to 600 to 1000, okay, in terms of concentration of sodium chloride. So we have a high concentration of sodium chloride at the bottom here, and as we turn the corner, what we start doing is we start moving sodium chloride ions out of here. We pump sodium chloride ions out. Okay, so the cells here have cells with lots of mitochondria producing ATP, which pumps sodium ions out of here, which reduces the concentration of sodium ions as we move up here. Okay, but the movement of the sodium chloride ions into this medulla region makes it more concentrated, more salt, higher salt concentration here, which reduces the water potential, which allows the water to move out of here. Okay, so it's quite a complicated process. So that's what happens in the descending limb and the ascending limbs of the loop of Henle. Descending limbs, water is removed. Ascending limb, sodium ions are actively pumped out. So the other thing, and the fact that the descending limb and the ascending limb are so close together creates what we call a hairpin countercurrent multiplier. And what that essentially means is that it's the active removal of the sodium ions in the descending limb actually creates a water potential gradient for water to move out of the descending limb. Okay, and that requires the descending limb and the ascending limb to be close together. In adaptations of kidneys of different mammals have different lengths of loops of Henle. Ones that are animals that live in very arid conditions, desert rats and so on, they tend to have very long loops of Henle, whereas mammals that live in an environment where there's lots of water, such as a beaver, they have very short loops of Henle. So, moving on. Once the filtrate has moved out of the loop of Henle, what then happens is it passes through the distal convoluted tubule, and you don't really have to know much about the distal convoluted tubule at all. In fact, you don't have to know anything at all about it, so we won't mention it. But we do have to know what goes on in the collecting duct. Okay, the collecting duct provides us an opportunity to regulate the amount of water that we absorb from our filtrate. So the filtrate passes from the ascending limb up through the distal convoluted tubule, then it passes through the collecting duct, which actually isn't a diagram of here, but it passes through the collecting duct, and then that eventually goes into the renal pelvis, which takes the urine into the ureter. The collecting duct is lined with cells which can alter their permeability to water. So if your water levels are low, the cells become more permeable, which allows more water to be reabsorbed, so we conserve water. If your water levels in your blood are high, what happens is that we make our cells of the collecting duct impermeable to water, so all the filtrate passes out into the renal pelvis and into the ureter, and we would make copious amounts of very dilute urine. Whereas when our water levels are low, we absorb more of the water, so we produce small amounts of very concentrated urine. The way it works is this. Just rearrange all of these for a second. If you can imagine that this structure here is a cell lining the collecting duct. When water concentrations are low, you produce from your pituitary gland a hormone called ADH. Okay, which just draws a little blob there. And ADH passes through the blood and binds to an ADH receptor cell. So this is cell signalling again. A receptor cell on the cell surface of the collecting duct cell. Inside the cell are vesicles which contain protein channels which allow water to pass through membranes. And these channels are called aquaporins. Okay, an aquaporin, so it's a water channel. So here are the water channels here and here. Now when they are in this vesicle, the cell membranes are not permeable to water. When ADH binds to this ADH receptor, what happens is a second messenger is produced in the cell, our old friend cyclic AMP. So there'll be adenyl cyclase here, which will convert ATP to cyclic AMP, a bit like we saw with adrenaline in the past. And what the cyclic AMP does is it causes the vesicle to move to the cell surface membrane. I can't actually do that in this little animation, but what does happen is that as the vesicle moves, these aquaporins are then inserted into the cell surface membranes of the cell. And what that means is that water can now pass through into the cytoplasm and out through the other side. So ADH, which was represented by this red blob here, causes the water channels to be transferred from inside the cell into the cell surface membranes, which makes this cell permeable to water, which means that we will imagine this was the filtrate here. So when that happens, ADH binds to the molecule and binds to the receptor. The aquaporins are inserted in the cell surface membranes. So water will start to move along a water potential gradient, okay, from the filtrate into the medulla. Because remember, the collecting duct passes through the medulla, and also because the water potential in the medulla is low because of all that sodium chloride in there, water will pass from the filtrate into the bloodstream, which will reduce the amount of water you lose in your kidney filtrate, i.e. you'll produce less amounts of urine and more concentrated urine. When the ADH isn't around, it isn't being produced, so ADH doesn't form there, the cyclic AMP isn't produced, what happens is that these water channels will then be corralled back into the vesicles in the cell cytoplasm, and the cell membrane will now be impermeable. Okay, so water will not be able to pass through here. So that's what happens when you've got lots and lots of water in your blood, is that no ADH is produced, therefore these cells here are waterproof, and therefore any water that gets this far in the nephron will just pass through in the filtrate, and go into the kidney. Go into the kidney pelvis or the renal pelvis. So you'll produce copious amounts of very dilute urine. The other thing you've got to be aware of here is the presence of how ADH is released. Okay, so we talked about what ADH does. ADH is produced actually by cells in the hypothalamus, okay, which is in the brain, and it's produced in special cells called neurosecretory cells, okay, whose cell body is in the hypothalamus, and they have an axon which stretches from the hypothalamus down to part of the pituitary gland called the posterior pituitary gland. Okay, and I'll just find a picture of that. Here we go. So if you can imagine this as a neurosecretory cell, here's the cell body of the neurosecretory cell, which is in the hypothalamus, and here's the axon of this neurosecretory cell, which stretches from the hypothalamus down to this structure, which is just above the roof of the mouth, called the posterior pituitary gland. So ADH, which Americans call vasopressin, is produced in the cell body, protein synthesis in action, so lots of ribosomes, endoplasmic reticulum, etc. It's then bundled into vesicles, which are then transported, okay, down the axon, which stretches all the way from the hypothalamus down to the pituitary gland, and then they're released by the pituitary gland. Okay, and there'll be special receptor cells called osmoreceptors, which will detect levels of water in the blood as it passes through the hypothalamus. So what will happen is that if water levels go down, that will send a message to here, which will stimulate ADH production. ADH gets transported along the axon into the posterior pituitary gland, where it's released into the blood, and then it circulates in the blood, where it binds to ADH receptors on collecting duct cells, which increases the water permeability of those collecting duct cells by inserting aquaporin molecules, which we talked about before, aquaporin molecules into the surface of the membranes. Now, finishing off our look at the kidney, there are various other little things that we've got to talk about, one of which is what happens when the kidneys fail. Obviously, if the kidneys fail, we've got a problem. We can't control water levels in our blood. We can't get rid of urea, which is would be poisonous when if it accumulated at high levels in the blood, and we can't control salt levels either. So somebody with kidney failure will become very ill very rapidly, and therefore there are various things that we can do with somebody who has kidney failure. Okay, and here are the various things that you need to know about. You need to know about something called renal dialysis, which you probably came across at GCSE. And there are two types of dialysis we can do, something called hemodialysis, which is probably the type of dialysis we encountered at GCSE, or something else called peritoneal dialysis, which I'll explain. And then there is the idea of kidney transplants. One of the first organs that were successfully transplanted were kidneys, and we can look at a bit about the issues surrounding kidney transplants. So first of all, let's look at dialysis. As I say, there's two types of dialysis. One type of dialysis involves blood being taken out of the patient and passed through a machine. Okay, and as it passes through a machine, the blood passes between membranes, and the membrane separates the blood from a substance called dialysis fluid. And the dialysis fluid contains all the right levels of things that should be in the blood, the right amount of glucose, the right amount of amino acids, the right amount of salts. And as the blood passes along, passes through the machine, passes through these membranes, what happens is diffusion occurs from the blood into the dialysing fluid, and similarly, some substances diffuse from the dialysing fluid into the blood. The idea being, by the time the blood gets to the end of this process, there will be the right levels of all the various things that are required in the blood. So if we just can watch a little bit of this video. Should be a video, let's see if it works. Perhaps not. Oh, here we go. So this is showing blood coming in. Here's the membrane that separates the dialysed fluid or dialysing fluid and the blood, and so substances pass between the blood and the dialysing fluid as it goes through the membrane, and the idea is that the blood coming out here will have all the right levels of glucose and amino acids and urea and so on. Urea will pass from here to here. If the glucose level's too high, glucose will pass into there. If glucose level's too low, glucose will pass from here to here. So that's the principle by which that works. Okay, I can show you that again. So that process there is known as hemodialysis. There is another type of dialysis that has become popular because this doesn't require equipment. What this actually uses, rather than an artificial membrane, it actually makes use of a large membrane that exists around our internal organs called the peritoneal membrane. The peritoneal membrane separates the abdominal wall from our internal organs. Okay, so at one side it wraps around all the intestines and various abdominal organs we have. And on the other side, it wraps around the outer, or the wall, the inner wall rather, of the abdomen. And there's a space between the two membranes, which is called the peritoneal cavity. And this membrane is supplied with blood vessels and so on. So if there would be an opportunity for blood molecules to pass through the membrane into fluid. So what we do very cleverly is we fill the peritoneal cavity of a patient with dialyzing fluid, and we leave it in there for a while so that equilibriums occur between the blood and the fluid that we put into the peritoneal cavity, and then we empty the fluid out and replace it, and do that a few times until we've managed to get rid of waste products and to equilibrate all the substances at the right levels within the blood. And I can show you a little video here of what happens.
PD uses your peritoneal membrane, the lining of your abdomen, as a filter. For this kind of dialysis, a tube, called a catheter, is surgically placed through the wall of your abdomen. This site is near the navel. About 3 to 4 inches of the catheter extends out of the body. This catheter is a permanent access for PD. During treatment, a special solution flows through the catheter into your peritoneal cavity. While the solution remains in your peritoneal cavity, waste products and excess fluid pass from the blood through the peritoneal membrane into the dialysis solution. Your peritoneal membrane acts as the filtering device, similar to the dialyzer in hemodialysis or to your own kidneys. After a certain number of hours, the solution is drained through the catheter and out of the peritoneal cavity. The used solution, which contains waste and excess fluid, is then replaced with fresh solution. You disconnect and discard the bags, and are now free to go about your day.
So the peritoneal dialysis has a few advantages. It doesn't need complicated equipment and expensive equipment, so sensible for National Health Service. It doesn't require you to find veins to put needles in, which becomes a problem after a while because you do it so often that your veins get so damaged, whereas peritoneal dialysis doesn't require that. So there's a lot of advantages with the peritoneal dialysis. One of the ultimate solutions to solving renal problems would be a kidney transplant. Kidneys were one of the first organs to be successfully transplanted into patients, and the kidney donor service, which gets kidneys from people that have sadly died through accidents and so on, can use those kidneys to be transplanted into people with kidney failure. One of the problems is tissue rejection, linked with F214, the immune system, is that obviously a kidney that doesn't belong to you will have antigens on it, which will be recognised by B cells and T cells, and an immune reaction will occur, and therefore after kidney transplant you need immunosuppressant drugs which suppress the immune system. That can be problematic because that can make you more prone to other diseases if you have a suppressed immune system. The other issue with kidney donors is a shortage of organs, and the whole issue, ethical issue, about whether people should be forced to carry kidney donor cards, or perhaps you should have an opt-out scheme whereby if you do get run over by a bus, your organs will be used unless you have a card which asks them specifically not to be used. So there are a few issues involved in kidney transplants. A few other things to consider with let's move some of these diagrams around, which I'll come and use later. A few other things to consider with the use of with the kidney. First of all, urine. A lot of substances are released in the urine, And sometimes it can be useful to find out whether these substances are there in the urine. Glucose is an interesting one. If you have high levels of blood glucose because you're a diabetic, some of that glucose will end up in the urine, and so testing urine for glucose is the first test for diabetes. Is an obvious one, but there are other tests that we can use. Pregnancy testing can be done by putting a test strip into a mother's urine, and what we're looking for in this case is a hormone which is released by a developing fetus called human chorionic gonadotrophin, or HCG for short. So a pregnant mother will produce HCG, and that will pass into her urine, and it can be tested for in the urine to discover whether she's pregnant or not. Okay, and this is rather cunning. It involves the HCG being just trying to rearrange some of these diagrams. Get those out of the way. So the first thing is, put those over there for a second. The first thing is that in the strip there are antibodies. Okay, remember these antibodies, these protein structures that look like a Y, an upside down Y in this case, which have regions that can specifically recognize proteins. These regions at the end here which can specifically recognize proteins. And these antibodies in this case are attached to a coloured dye molecule, coloured bead we'll call it. Okay, that's a dye molecule, okay, which is a particular colour. And these antibodies are specific to the HCG hormone. So imagine you are pregnant, the HCG will bind to our antibody. The HCG antibody coloured bead complex will then, so here's a HCG antibody coloured bead complex, okay, we've got a few of them here. So those will then pass up through the strip by capillary action. They'll pass up through some inert material on the strip, where they will then encounter some what we call immobilized antibodies. These are antibodies which will be specific to the HCG, but what they are bound or immobilized to some inert substance, okay, which forms a strip across your test strip. And what happens is the HCG antibody bead complexes bind to the HCG like that to form this even more complex thing, which we can call an HCG antibody coloured bead, passes up the test strip and encounters a region where further immobilized antibodies are present. This forms an immobilized antibody, HCG antibody coloured bead complex. So immobilized antibody, being this structure here, then binds to this structure here. And what it does is all the coloured beads, or dye molecules, then form a nice neat line here, which one can actually visibly see as a nice neat line across this test strip, which means you're pregnant. Okay. Whereas if you're not pregnant, there wouldn't be any HCG molecules to bind to my antibody with its coloured bead. Okay, and as as this passes through, okay, it wouldn't have anything to bind to the immobilised antibody, and these would just pass on straight through and wouldn't form a coloured strip along here. Okay, so that's how pregnancy testing works. So this is a test strip here with immobilised antibody. Hope that makes sense. The other thing we can test for, okay, and we do this at Marlborough College, we can test for drugs in urine. Okay, if you go ask for a drugs test, you have to go to the sanatorium and they take a urine sample. It goes off to a laboratory where they test the urine for substances. This is also involved in sports bodies. Will test for anabolic steroids, which cause rapid muscle growth, which are illegal in sport. So testing will occur. The testing is the principle by which this testing happens is similar to our own drugs testing that we use for recreational drugs, and essentially what we do is that we take samples of urine and we analyse that by a process known as gas chromatography. Okay, and gas chromatography is when we boil our urine, or boil all the molecules in the urine, okay, and pass it along a tube which has attached to the inside of it a substance which has a liquid, a high boiling point liquid attached to it. And as the gas, vaporized urine, passes along the tube, what happens is some of the molecules dissolve in the liquid that lines the tube, and different substances will dissolve at different rates into the side of the tube, and as a result it takes different molecules different lengths of time to pass along the tube. So different molecules will emerge from the other side of the gas chromatography machinery at different times, and that can tell us what the substances are. Again, I have a little video which illustrates what's going on here. Let's have a look at this. So I'll show you that again in a minute. So what we did here was that we have our sample of urine. Okay, we then inject it into the machine where it's vaporized. Okay, and it passes through this column. Okay, and this column is in an oven. Okay, so that it's very hot in here. But clinging to the side of this tube, okay, is some sort of inert material, inert solid which has attached to it a liquid, okay, a high boiling point liquid. Okay, so this is liquid even though it's in an oven, okay, because it's got a high melting point. And what happens is that the molecules, what they do is they dissolve in the liquid as they're passing along. Okay, and they sort of come in and out of solution as they're passing along. So different molecules will take different lengths of time to pass through this tube. Okay, and then as the molecules emerge, they're detected. Okay, they're detected. So different molecules will have a different time that they pass out. So an anabolic steroid will have a certain amount of time that it will take to pass through this particular chromatography machine. And as a result, we can analyse the contents of the urine, which molecules are involved. Okay, so I'll just show you that again. So the molecule comes up here, mixture of molecules which are separated because they dissolve in the liquid at different rates, and they eventually come out separately, and they give you this trace because they all come out at different times, and the time indicates what the actual molecule is on this trace here. So these represent different molecules because we know that different molecules take a certain amount of time to pass through this particular column. So let's imagine this was an anabolic steroid. The time it took for an anabolic steroid to get through this machine. If we passed some urine through this and we got a spike there, we would know that the athlete was taking an anabolic steroid.